All right, folks, let's go ahead and get started. This is the VCU Internal Medicine Grand Rounds. Today is September 12th, 2024. Just a couple of quick housekeeping items. We are here in the Molecular Medicine Research Building on the first floor and also online every Thursday, 12 to 1 o'clock. For those in person here, just want to remind folks, maybe mute your cell phones if you can. And the CE code is now on the screen. It will also be displayed at the end of the lecture for people who need to record their attendance, for either house staff or faculty members who need the CE credit. Today, we are honored to bring in Dr. Aron Lichtman. Dr. Lichtman is going to focus on endocannabinoids, building targets to treat pain. Dr. Lichtman earned his Bachelor of Arts in Psychology at Rutgers College and proceeded to earn his Ph.D. in Psychology at Dartmouth. He then received postdoctoral training in Pharmacology and joined the faculty here at VCU in 1993. Has risen through the ranks to full professor. He served as the Associate Dean for Research and Graduate Studies in the VCU School of Pharmacy from 2015 to 2023. His research program focuses largely on cannabinoids, and he has been continuously funded by the National Institutes of Health for more than 30 years. The International Cannabinoid Research Society recognized Dr. Lichtman for outstanding research contributions with the 2013 Michaela Award, and he's previously served as president of that organization and recently elected to serve on the board of directors of the College on Problems of Drug Dependence. He also is a co-inventor on 3 patents, 2 of which include a THC metered dose inhaler that progressed through phase 2 clinical trials. In addition to his scholarship, Dr. Lichtman is a dedicated teacher and mentor and has trained 17 PhD students, 13 postdoc fellows, and many high school, undergraduate, medical students, and post-baccalaureate students. He twice received the Professor of Year Award from the Department of Pharmacology and Toxicology, and the VCU School of Medicine recognized him with the Distinguished Mentor Award in 2013. Dr. Lichtman's research focuses on drug addiction, pain, and other diseases of the central nervous system, with an emphasis on the endogenous cannabinoid system. He was invited here to speak to us about some of his recent projects, as well as talking about new targets to treat pain and exploring the endocannabinoid regulating enzymes. Dr. Lichtman, please come up and share your thoughts.
Well, thank you, Dr. Bergman. And I want to thank Dr. Bergman and Dr. Carr for the kind invitation. And just to go through an overview, I'm going to start just really by talking a little bit about the plant, on the background of the discovery of the active constituents, namely THC and CBD. And then I'm going to go through very rapidly the discovery of the endogenous cannabinoid system. And this is very analogous to the discovery of the endogenous opioid system, which really occurred back in the 1970s. With just a few differences that we'll get to. So we'll start with the plant. We'll go to these active constituents here that everybody, I think, is familiar with. And we'll talk about the approved therapeutic uses as well as the potential therapeutic uses of cannabinoids. And then I'll go through, in kind of broadly, the endogenous cannabinoid system. And this just shows the expression of different components of the endocannabinoid system on different cell types within the central nervous system. And we're not going to get into that much detail. And then finally, I'll talk about how at least one of these targets, an enzyme that metabolizes one of our predominant endocannabinoids, is a potential therapeutic target for treatment of pain. And it may also be involved in some of the pathology with sickle cell disease. And I want to just start by saying that this project really is the culmination of, some work that was done initially by an undergraduate working with me and brought me in close touch with one of the faculty in internal medicine, Wally Smith, who, is a renowned scientist in sickle cell disease. So I learned a lot from Wally, and we, put a few things together And now we're getting some funding for this project and moving forward in terms of science. So let's go ahead. I think we are familiar with the learning objectives here. And I just want to start by discussing, cannabis. It's been used medicinally and potentially recreationally for thousands and thousands of years. There is archaeological evidence that it was used medicinally for treatment of pain, in the Mideast, dating back about 5,000 years. Same thing in China. In some of the ancient textbooks, it lists cannabis as a medicinal plant. Same thing through ancient Persia. Today's Iran lists cannabis as a medication. And a lot of the same reasons why people are using it today, from dispensaries for treatment of pain, for seizure disorders, for anxiety, the ancients knew about this as well. So we haven't really progressed that far. But it's taken on a lot within society. And it's just, another plant that has chemicals, substituents that have medicinal properties. So, here are a few other plants that have that with maybe a little bit less controversy. So I think we know what makes cannabis unique, and it is the delta-9 tetrahydrocannabinol, THC, which is a agonist at cannabinoid receptors, and it's really responsible for the psychomimetic effects that we associate cannabis with. But in addition to THC, there's about 120 other structurally similar molecules, and collectively they're known as cannabinoids. And we'll touch just upon two of those. And I also want to go through this list down here, and we have something called terpenes, and I think the list is much higher now. And that's what gives cannabis a unique smell, that it has a pungent odor, you can't miss it. And there's also a lot of interest in discovering potential medicinal uses of the minor phytocannabinoids as well as the terpenes. So there are 3 FDA-approved medications that are cannabinoid-based, and potentially there's a 4th that I'll talk about as well, very briefly. The first one that was discovered was the active ingredient in THC, that is called THC. That was discovered about 60 years ago by Raphael Mechoulam, and it was approved in the 1980s as an antiemetic and for antiemesis, for cancer-related nausea and vomiting. And it's still used, and then a few years later, it was approved as an appetite stimulant for AIDS-related cachexia. Some of its off-label prescriptions are for stimulation of appetite for cachexic cancer patients as well. So these are the approved uses. It has a lot of other potential uses in addition to these. The second approved medication is a synthetic cannabinoid, and this drug is called nabilone. It's about 10-fold more potent at the receptor than delta-9 THC. And again, it is approved as an antiemetic for cancer, chemotherapy-induced nausea and vomiting. Side effects include, sleep-inducing and all the psychomimetic effects that cannabis has. The third FDA-approved medication is Epidiolex, or that's cannabidiol. And cannabidiol is very interesting. Structurally, it's very similar to delta-9 THC, except this ring right here is open and it's hydroxylated. As a result, it loses psychoactivity that delta-9 THC has, but it has anti-epileptic properties. So the same individual who discovered THC 60 years ago published the structure of cannabidiol actually 61 years ago. And and that was Mechoulam, and they had done some clinical trials in the 1980s, early 1980s, showing it had anti-epileptic properties. And that was after showing that CBD reduced convulsive behavior and seizures in different rodent models of epilepsy. So today, CBD is approved for these severe seizure disorders, They're taken as an adjunct with other anti-epileptics. Mechanism is largely unknown of CBD. And happy to talk about it later if there are questions, but it's not going to come up too much more in this talk. So in addition to potential therapeutic uses, there are some concerns with cannabis-based drugs. And particularly in this day and age where we have medical dispensaries and recreational use and people can grow it, and there's a general feeling among individuals that cannabis is entirely safe. As a result, much of its use has increased a lot. So whereas nobody's ever died from the direct effects of cannabis or THC, it does have some untoward effects. There are thousands of emergency room visits each year because of cannabis. And a lot of this has to do with the high potent strains that are available, or even the synthetic types of cannabis that are there. Actually, it's not synthetic cannabis, it's synthetic cannabinoids, which some of them are about 100-fold more potent than THC. And that was a much bigger problem about 10 years ago, but they're still out there. There's also a lot of accidental overdoses or poisonings with the edibles. So a lot of the dispensaries and even at recreational shops, you could buy it as candy. And especially young children going through their parents' stash, they'll see this as candy and they'll take too much. And, same thing with adults. A cookie could have a lot of THC in it, and it's advised, you only take, a quarter of the cookie. And due to its pharmacokinetics, taken orally, it takes about an hour to 2 to get effects. A lot of times people, hey, I felt no effect, and they finish the whole cookie and they've gotten way too much. And just, you can go through this list yourself of some of the concerns. There is a dependence phenomenon with it. It is associated with schizophrenia. It does impair driving. It does increase accidents overall. So it's a pharmacological agent, just like many other drugs. It can do positive things and also have some negative things as well. So once THC was discovered, it gave us some tools to try to understand its mechanism of action. And there are 2 cannabinoid receptors. These are G protein coupled receptors. They're termed CB1 and CB2. The CB1 receptor is responsible for most of the pharmacological effects that we associate cannabis with. It is the most highly expressed GPCR in the brain, and it's largely expressed presynaptically. So its stimulation regulates neurotransmitter release, particularly GABA, which is inhibitory, or glutamate, which is excitatory. There are potential therapeutic effects by CB1 stimulation and some of the side effects that I've already discussed. The second cannabinoid receptor, creatively called the CB2 receptor, has about 60% homology of the CB1 receptor. It's somewhat truncated. It has higher homology within the transmembrane domain. But unlike the CB1 receptor, which is highly expressed on neurons and other cell types as well, the CB2 receptor is largely restricted to immune cells. So within particularly microglia, macrophages, B cells. Stimulation of the CB2 receptor in at least laboratory animal models produces analgesia and anti-inflammatory effects. And it doesn't have any apparent adverse side effects at this receptor. So medicinal chemists, basically tweak the molecule, delta-9-THC, and were able to design drugs that selectively bound to CB2 receptors. Many of these have been through clinical trials, but generally there's been a lack of efficacy. So we have the existence of cannabinoid receptors. So I told you earlier that the story is very analogous to the story of endogenous opioids. So why did we evolve to have these receptors? So likewise, opiate receptors were first discovered by Saul Schneider's group up at Johns Hopkins. And then soon after that, beta endorphin and then enkephalins were discovered. And the same pattern of effects of discoveries occurred in the case of the endocannabinoid system. So no, we haven't evolved to have cannabinoid receptors just in the off chance that our forefathers and mothers would come across a cannabis field. But the plant material, that is a delta-9 THC, was mimicking what's naturally in the brain. And the same individual, Raphael Mechoulam, who discovered or saw the structures of THC and CBD, was largely behind the discovery of the first two endocannabinoids, anandamide and 2-arachidonoglycerol, or 2-AG for short. Very interesting story. And these are fatty acid amides. These are lipid signaling molecules from distinct families of lipid signaling molecules that happen to bind to the CB1 and CB2 receptor. They look nothing like delta-9 THC, but they do bind to the receptors, as I said, and activate them. And the thing is, when we first started to work with these lipids, they were really hard to study and do any kind of pharmacological experiments with because they would be degraded so rapidly. And that's because they're basically produced on demand by biosynthetic enzymes and then rapidly degraded by other sets of catabolic enzymes. So this just shows the major enzymes for 2-AG and anandamide. And 2-AG, it's a little bit better worked out. We have these diacylglycerol lipases that play a role in 2-AG regulation. They're really interesting just by themselves because they do play a role in inflammation and many different physiological processes within the central nervous system. And, same thing, we've got several different biosynthetic enzymes for anandamide, and we have two major catabolic enzymes for each of these. One of them called fatty acid amide hydrolase, FAF is short for anandamide, and the other one is monoacylglycerol lipase, or MAG lipase for short, that's for 2-AG. And I will be talking a lot more about 2-AG for the work that I've been doing with Wally Smith. So a little bit more about that. And also it's very relevant that they're then broken down by these enzymes to either glycerol or ethanolamine and arachidonic acid. And that arachidonic acid becomes very relevant when we talk about MAG lipase inhibition. And this is just an interesting fact. Acetaminophen, anybody know the mechanism of action for acetaminophen? Largely unknown. It's not a COX inhibitor, but there's a lot of evidence suggesting that it is metabolized, to first para-aminophenol, and then to this chemical called AM404, that was independently synthesized by an investigator at Northeastern University that activates the endocannabinoid system. So he made this stuff synthetically, but it turns out, rodents and humans will synthesize AM404, and hasn't been proven yet, but it's very likely that this metabolite, so you could think of acetaminophen acting as a prodrug, is contributing to some or most of acetaminophen's therapeutic effects. And that's interacting with the endogenous cannabinoid system too. It interacts with that enzyme fatty acid amide hydrolase, or FA. Okay, so to move forward, I want to talk a little bit more about 2-AG. And just in the interest of time, I'm not going to get all into the biosynthesis. These are very interesting enzymes. And what it tells us is that the 2-AG is being manufactured in different types of cells, and there are different stimuli that cause that. So again, when I talk about 2-AG and endocannabinoids being produced and released on demand, there are stimuli out there that cause that. So it could be stress that we could manipulate, or pain that we can manipulate. But within the central nervous system, there are chemicals that are then causing the release of and the production of 2-AG. The enzyme of interest here is monoacylglycerol lipase. That's the major metabolic enzyme for 2-AG. And it doesn't only metabolize 2-AG, it metabolizes other arachidonoglycerols. But very importantly, it's not only a catabolic inhibitor, it's also a rate-limiting enzyme for the production of arachidonic acid in certain tissues, not all. So particularly within the brain, within neurons, within liver and lung, but not in heart, kidney, spleen, or gut. So when you inhibit 2-AG, and this is what I'll be going through very soon, you get, when you inhibit MAG lipase, you get an increase of 2-AG, which then stimulates CB1 and CB2 receptors. And you also get a decrease in free arachidonic acid. And less arachidonic acid, there are a number of enzymes that work on that, such as cyclooxygenase, and there are less prostaglandins. So you have multiple mechanisms of action for analgesic and anti-inflammatory effects with a MAG lipase inhibitor. So as I said, we're going to really focus on MAG lipase here. And let's move forward. I kind of gave this review already. The potential therapeutic indications of the endocannabinoid system. In red are FDA-approved uses. In black text here is everything else. And, there is a good deal of evidence suggesting that cannabinoids have mild to moderate effectiveness with some types of pain in some patients. But again, this is not FDA approved. Likewise, THC cannabis is reported by some individuals to reduce anxiety, but too much of this stuff will provoke anxiety. And that's really the cause of a lot of the emergency room visits. So I'm going to focus on sickle cell disease. And that is, there's a lot of work that's already implicated the use of cannabis in patients who are suffering from pain in sickle cell disease. And there are also rodent models for sickle cell disease. And, again, this is largely, this is hereditary disorder that initially came from Africa and also around the Mediterranean. We also have a lot of individuals within the US who suffer from sickle cell, and it is associated with severe pain, with organ failure. The sickle cell crises really require hospitalization. And this generally is a disease of underserved individuals, and there's also disparities in research of sickle cell disease. But fortunately, there's been a lot of research lately. Okay, so this, basically this shows that it occurs as a result of a mutation on the beta globin gene on chromosome 11. And it causes the hemoglobin subunits to polymerize to form long rigid molecules. And as you can see, they sickle basically, and it could cause occlusions within the blood vessels that could lead to ischemia. And on the right, it shows a pediatric patient with severe inflammation, and you could just see the look on this child's face of severe pain. So there really isn't good pain management for this. NSAIDs really have minimal effect. Some of the weaker opioids are used, and also oxycodone, morphine can be used as well. But all of these come with side effects, and there's also stigma associated with the opioids. So sickle cell patients have also reported to use cannabis to manage their pain. And there are a lot of reasons for it, and there's a lot of self-report and case studies on it. But a study done by Don Abrams just several years ago looked at vaporized cannabis, and they did not find that it significantly reduced pain in a sickle cell patient. But it did improve mood and had minimal adverse effects in them. So, a lot of this has to do, with dose response effects and the severity of the pain and other individual components. There's now, if we kind of switch gears a little bit, and that's talking about a mutant mouse model of sickle cell disease. in which the mice have been mutated to express the human sickle cell gene. Different synthetic cannabinoids have been shown to reduce nociception in various pain models in mice. And also some of the enzyme inhibitors, such as Pha, the one I mentioned before, also had some moderate success in reducing the increased nociception in the sickle cell mice. There's also some other interesting evidence that implicates 2-AG in sickle cell disease. So in the mice, it turns out that 2-AG is reduced and prostaglandins are increased in the dorsal root ganglia of various types of sickle cell mice. And this is consistent with an increase in expression of MAG lipase. That's what you would expect. There are other ways you can get there too, but increased MAG lipase would reduce 2-AG and increase the arachidonic acid. And also inhibiting 2-AG biosynthesis has been found to also reduce the pain responses in the mouse model. So let's just kind of jump to the hypothesis here. And in the terms of sickle cell, either the crisis or after a chronic condition of the sickling, we hypothesize that MAG lipase is increased. It's upregulated. As a result, there would be increased degradation of 2-AG, less 2-AG, less stimulation of CB1 and CB2 receptors. And if we're increasing the catabolic enzyme, we're going to be increasing its primary metabolite, arachidonic acid. And as a result, there are other enzymes that act upon arachidonic acid, particularly cyclooxygenase. So there would be an increase of prostaglandins, and that's been the case. So we propose that a MAG lipase inhibitor would basically normalize this system, that we would get normalized or actually elevated levels of 2-AG, further stimulation of CB1 and CB2, and then a decrease of arachidonic acid, and less arachidonic acid will lead to a reduction of prostaglandins and other of its pro-inflammatory metabolites. So that's where we got started. But first, we examined the sickle cell mice, and we looked at the spinal cords and dorsal root ganglia of these animals, and looked at various components of the endocannabinoid system. So this work was done by Dr. Brian McIver, who is now a postdoc at Yale, but he earned his Ph.D. here in pharmacology in Dr. Damage's laboratory. And as a student, he was very curious. He knew we had sickle cell mice. And he said, I'd like to look at, opioid receptors and different components of the endorphins, the enkephalins, the peptides. And do you have some tissue available? I said, sure, but can you also look at components of the endocannabinoid system? So he found some very interesting results. And using this, I told you about the human sickle cell mouse model. This is a Berkeley mouse model. And there are controls. These are called HBAA Berkeley mice. They're in a mixed background, but they express a normal human hemoglobin gene. So no pain for them. Those are controls. And the humanized sickle cell mice are the experimental group. So what we looked at was 5 versus 10-month aged mice, because it's been shown by us and others that as the animals age, the chronic pain becomes more severe. And I kind of jump ahead. I'm showing you the MAG lipase. And Brian did PCR. And I'm just showing you on the left is the dorsal root ganglia data. On the right is spinal cord. And you can see all the effects are happening in these peripheral neurons in the dorsal root ganglia, just outside roughly, I think it's about T4 to L6 of the spinal column. And there is an increase of MAG lipase in the sickle cell mice at 5 months of age, and there's a further increase about a threefold increase compared to controls at 10 months. And at this point, the animals are showing a lot of severe signs and increased sensitivity to touch, to heat, to cold. That is, they're more in pain. And they're showing a lot of functional deficits as well. We, real quickly, we looked at the biosynthetic enzymes of 2-AG, no real changes for both isoforms of that. And then Brian also looked at various pro-inflammatory cytokines. And as you could see, IL-1 beta is way up in the dorsal root ganglia of the aged mice, but not the mice that are 5 months of age. And this is also reflected in the spinal cords. IL-6 was also significantly increased in the dorsal root ganglia as well as in the spinal cords, but it wasn't so much age-related. But also, not all the cytokines were increased. TNF-alpha, there was no genotype effect on TNF-alpha, so not everything is going up. But it does show us that there is a dysregulation of MAG lipase, which degrades the endocannabinoid 2-AG. So next, we examined an inhibitor of MAG lipase in different pain assays in the sickle cell mice and the controls. And we also looked at functional behavior. And I'll go through those data right after I kind of go through the methods here. So in this study, we took male and female control mice, and as well as the humanized sickle cell mice, about 5 months of age, where they show a pretty good pain phenotype. The MAG lipase inhibitor we used was a drug called MJN110. It's highly selective. It's very potent. These are the doses that we used. And the animals were tested repeatedly in a Latin square design with about a 3 or 4 day washout period to prevent carryover effects. The primary dependent measures are stimulus-evoked nociception using von Frey filaments. So basically, increased sensitivity to touch, which would be a mechanical allodynia. And that's right here, the von Frey. We also used hot plate for thermal hyperalgesia. And what do we have here? On our left, it shows the von Frey data. Y-axis shows the stimulus intensity, the number of grams. On our x-axis, it shows the control mice, HBAA here, versus the sickle cell mice on the right in red, and the dose of the MAG lipase inhibitor. So the control mice really will move. So basically, the animals are on a screen with a plexiglass enclosure around them, and the experimenter takes the von Frey filaments and just pokes at the bottom of their feet, their paws, that is. And a withdrawal response is measured as the endpoint for nociception. So the HBA mice takes about 1 or 2 grams before they move their paw, and MJN110 has no effect on that whatsoever. The Berkeley sickle cell mice are severely allodenic, so they are showing a robust withdrawal response to these very thin filaments, about 1/10 of a gram. And as you could see, the MAG lipase inhibitor dose-dependently reverses this pain response. We get a little bit of a different story with heat sensitivity. And although we had some significant effects, it was only a partial reversal. So big point here is these animals show a lot of different sensitivities to touch, to heat, to cold. And the MAG lipase inhibitor was very effective with touch, not so effective with heat. And I'm not showing you the cold data because the cold sensitivity doesn't develop until the mice are about 10 months of age. We also looked at the animals in a battery of functional behavioral tests. And I'm only going to show you 2 right here. One which we found was sensitive to a MAG lipase inhibitor and the other wasn't. So first we looked at grip strength. So the mice hold on to this to this contraption, and you could measure the amount of force that they hold on to before they let go. So some have interpreted these results to be a matter of deep tissue pain. Whatever the reason, the Berkeley sickle cell mice show about a 50% reduced strength compared to controls, and it makes sense. The other is nesting behavior. Mice are natural nesters. You put them in a cage with these little nestlets, and over a period of a couple hours, they will tear them up and build a little nest, like right here. And it could be scored, and you could then have these two different measures for functional behavioral deficits. So let's take a look first at grip strength on your left right here. And again, everything is set up the same. On our y-axis is grams of force, and that's how hard the animals could hold on to their grip strength is. The control mice have a strength of about 100 grams they could hold on, and the MAG lipase inhibitor had no effect on that. The sickle cell mice had about half the strength of the control mice. And the MAG lipase inhibitor, in a dose-dependent fashion again, reversed this deficit in grip strength. All that looked well and good. What about nestlet consolidation? So the data were transformed to percent effect. And that's basically 100% means they built a beautiful nest, and 0% means that they didn't touch the nestlets. And as you could see, the control mice over the test period built very nice nests, and the sickle cell mice showed severe deficits in it, at least during that time point. And the MAG lipase inhibitor did not reverse this at all. So the deficits were still there. So I want to show one last, actually there are two more sets of data, but this one is very relevant. This assay is used to infer an internal affective state in the mice. And this is called the condition place preference paradigm. And this is basically the reason for using this is we can make inferences, possibly whether the animals are in an internal aversive state or not. And let me just kind of explain how this assay works. The animals are put in a 3-compartment chamber. There's a neutral one, and then on the left and the right, and this could be counterbalanced, they're given either vehicle or drug. And they get several pairings for this. It's very much of a Pavlovian type of paradigm. And initially, this was developed to look at rewarding properties of drugs. So if you give the animals a drug like cocaine or morphine or heroin or anything that has abuse liability, they'll develop a preference for the drug side. So during conditioning, they either get vehicle, they're only allowed to be in the vehicle compartment, or when they're given drug, they're in the drug compartment. If we gave them these other drugs of abuse, you would do it the same way, and the mice would show a preference for the drug side. And that is interpreted as, hey, it has potential abuse liability. So we use this assay. We could look at that potential abuse liability, but we could also look in animal and chronic pain. And this has been done with many other pain assays, nerve injury, with arthritic models, with cancer models, with chemotherapeutic models. And we also looked in the sickle cell mouse. And the idea is if sickle cell mouse is in this chamber, and if they're given the MAG lipase inhibitor, would they show a preference for that side? And then again, the inference would be that it's reducing some internal aversive state. I'm avoiding saying pain, but that's really the implication here. In the control animals that we have, those animals are not in a pain state. So if they show a preference for the drug side, that would tell us our drug, our MAG lipase inhibitor, has potential abuse liability. Any questions? Okay, so what did we get? So over here, let me just kind of orient you to the data. On our y-axis, that's preference for the drug side over the vehicle side. We have our control mice, HBAA. We have our sickle cell mice, HBSS. And white bars are the animals that got treated with vehicle. And the black bars are the animals that got treated with an effective dose of the MAG lipase inhibitor, MJN-110. And as you could see in the control animals, MJN-110 did not produce a place preference. The animals had no preference over the drug compartment or the compartment that was associated with vehicle injections. And I did not mention before that during the tests, the animals are not under the influence of the drug. The drug is washed out. So we're just looking at a pure preference or their memory. Now, when you look at the sickle cell mice, the control animals really don't have a preference for one side or the other, but the animals given the MAG lipase inhibitor have a very strong preference for the drug side, the MAG lipase inhibitor side. So this condition place preference gives us the inference, or what we infer from that is that there is reduction in some sort of internal aversive state. And then in the last study, because pain is often a chronic condition in sickle cell disease and also in the mouse model, those animals are just chronically hypersensitive to touch. Patients would need to get the drug repeatedly. And it's very important to show or to know whether or not tolerance occurs or would the drug continue to have its analgesic effects. So we looked at different doses of the MAG lipase inhibitor. This was all done in the sickle cell mouse. And again, same thing on the y-axis. And this dotted line would show where the control animals would lie. And this is after 7-day treatment. And after 7 days, the high dose of the MAG lipase inhibitor, it retains its antinociceptive effects. So there's no tolerance here. And with the low dose, we don't get any changes. We don't get an increase of effects. So, of course, it would be important to look at more chronic dosing and looking in other models as well. So what I showed you here is the Berkeley sickle cell mouse model shows an age-related increase of the endocannabinoid regulating enzyme MAG lipase in dorsal root ganglia. There's also an age-related increase of the cytokine IL-1 beta. And we also just went over some data showing that a MAG lipase inhibitor reduces some, but not all, of the pain phenotype of these mice. And the condition place preference study I showed you, that shows that it probably lacks abuse liability because it did not produce a condition place preference paradigm in the humanized control mice, but it did produce a condition place preference in the Berkeley sickle cell mice, again, meaning that it looks like it's reducing this internal state of pain. So where are we going from here? As I said, we really need to look at chronic administration of a MAG lipase inhibitor. Will we get sustained antinociceptive and anti-inflammatory effects in a much larger battery and after more chronic treatment? We also had done some studies, but I didn't show you for the sake of time today, of mechanism of action. And it's kind of interesting. Remember, I told you, you block MAG lipase, you get increased 2-AG, which could signal through CB1 and CB2 receptors. So we've looked at CB1, CB2 involvement. And both of the receptors are involved in the von Frey response, the allodynia, the mechanical sensitivity. But only CB2 receptors seem to mediate the beneficial effects that the inhibitor has on grip strength. In the future, we want to look at a peripherally restricted MAG lipase inhibitor. So given that sickle cell is largely peripheral disease, although it certainly has effects in the central nervous system, it could be that a lot of the pain and the pain-reducing effects of the MAG lipase inhibitor are happening more locally, maybe in the dorsal root ganglia, maybe in other sensory nerves. Maybe it can offer some benefit within the blood itself. So there is a peripherally restricted inhibitor. And then Wally Smith and I received at the beginning of the summer an internal grant from VCU, a breakthrough grant, to examine whether MAG lipase is upregulated in sickle cell patients and if that is associated with chronic pain or the degree of pain in those patients. And then likewise, we're looking in the mouse model as well. So at this point, we've only looked at dorsal root ganglia, but we're going to look directly in blood. And other work has shown that MAG lipase is expressed within the red blood cells. So this is, we're going through the whole IRB process and we're trying to get our animal colony together. So it probably won't be till next spring until we start to get some information about this. We also submitted an R01 looking at whether MAG lipase is a potential target to treat sickle cell pain. So we're going to use some of the, assuming we're going to have to resubmit the R01, hopefully we'll have strong pilot data that implicate this, but we'll see. Finally, there is a MAG lipase inhibitor that's gone through some clinical trials, and it's made it to phase 2 trials for Tourette syndrome. There was some old data suggesting that cannabis and THC is effective of reducing some of the tics and utterances in Tourette patients. It was a very nice idea. It didn't pan out. So the phase 2 trials ended. But the good news is this MAG lipase inhibitor, ABX 1431, was found to be safe, and even after chronic use. So there is the potential for clinical trials. That would be a long way down the road. So finally, I just want to acknowledge just a few people here. This woman in the middle is Kennedy Goldsborough, Dr. Goldsborough. She's now doing her postdoc at Emory doing more sickle cell research. And she's the one who connected me with Wally Smith and really during COVID, early in COVID, took the lead in doing a lot of these studies. She had recruited a couple of these undergraduates, and one of them on the right is Sarah Hers. She's now in our Ph.D. program here at VCU with the Damage Lab, and Ato Heni is working at NIH this year, and he's applying for MD Ph.D. programs. So, again, VCU, great place to collaborate. And this has been a really fun project because it includes basic scientists with different backgrounds and physician scientists. So at this point, I'd be happy to pause and answer any questions that you may have.
Thank you. Thank you so much. Any questions, comments? Yeah.
Can people assume maybe by us?
We'll have to repeat the question.
Yeah. Well, first of all, thank you very much for taking the time and talking to us about my question is, does MAG lipase or indirect AG is associated with psychodynamic effects of THC and activating the CB1 receptor, or is it not? I mean, the reassuring thing is not, it looks like it does not at least have the potential of misuse. But I'm just curious if it's associated with the psychedelic effects.
Yeah, that's a great question. So it has been studied both in humans. Oh, yeah, I'm sorry. So the question is, does a MAG lipase inhibitor that elevates to AG, does the inhibitor produce psychomimetic effects similar to cannabis and delta-9 THC? So, and the answer is it's been studied both in laboratory animals, particularly mice, and also in humans. So the human data with high doses, people indicate they felt something a little bit funny. They didn't report, there's different instruments that's used to measure cannabis intoxication. They didn't report being high. They didn't report being sleepy so much, but they felt something. The mice, for the most part, show normal cage behavior, but there are very specific assays that we could use to look at internal subjective states. And in those conditions, those animals, given a high dose of a MAG lipase inhibitor, will tell us that it feels a little bit like THC. So your answer is, I guess the answer to your question is a little bit. It is stimulating the CB1 receptor, certainly not to the degree that cannabis would or taking THC orally. The other difference with the inhibitor is even though we're going to be elevating levels and we could measure that, a lot of it isn't necessarily signaling. So the idea is you're going to be getting release in specific circuits of the endocannabinoid. So even though you may be inhibiting the enzyme, you're not just flooding the receptors everywhere. And so that's one of the advantages of having a drug that's targeting the enzymes as opposed to just globally hitting the receptors. Thanks for that question.
Yes, so I have a clinical question. Patients who use cannabis, they obviously claim for all anti-nausea effects. One of the things that we see as a patient is hyperemesis, nausea, vomit, and it's probably become an epidemic. So what do you propose in the reason?
Okay, so the question was, although THC is approved as an antiemetic for cancer chemotherapy, and patients have reported that, hey, it reduces my nausea, other patients, one of the side effects, report hyperemesis. So that's a known side effect. So what's the cause of that? And I've heard from psychiatrists who were treating patients who were long-term cannabis users. So Jim Levinson, who is he in internal medicine? Psychiatry, sorry. I usually end up learning, he comes to me for questions about, cannabinoids, cannabis, and I usually end up learning a lot more from him than he learns from me. So he had a patient that had been using it in his 60s for a long time, no problem, and all of a sudden developed this. So the answer really is the circuitry is very complex. The CB1 receptors, as I said, are expressed presynaptically on glutamate and GABAergic neurons. And so therefore, there could be a lot of differences that are related to dose. Are you inhibiting an inhibitory system? Or are you inhibiting an excitatory system? And you're inhibiting multiple excitatory inhibitory systems. And so, the real answer is we don't really know, but my guess is some of it could be dose related, and there could be adaptations over the years where all of a sudden there could be a downregulation of the receptor and this other effect appears, but it's well described and it's a very unpleasant side effect.
In patients who are practicing, these people end up in the ER. Vomiting, but down with pain. And they end up with CTs and ultrasound, blood tests, and they're off the hospital. So it's a big issue, and I don't know the reason. Something that maybe we can talk about and see whether we can look at. The other question is, you mentioned the cannabinoid inhibitor was used in fluids. Where exactly? Can you tell me where we used much of the patient?
Okay, so the question was about the MAG lipase inhibitors and the clinical trials and where were they conducted and what type of individuals they use. So this was done by Abide Therapeutics, and they were since then taken over by another drug company. And they used, in their initial studies for doing phase 1 safety testing, they decided to use patients who had, or individuals that had Tourette's, with the idea of, let's see if there's a potential therapeutic signal. So again, everything was safe with it. And as it usually goes with phase 1 studies or open label trials, boy, it looks good, that placebo effect or you're underpowered. But when they did the double-blind study for phase 2, it failed for Tourette's. So, and I believe it was done in a number of different centers throughout the world, not just the US. It was outside of the US as well.
Have these sectors, I'm not sure they have research. Have they looked into this in chronic pain syndrome? The sectors, the receptors, the characteristics in pain, for example, in pancreatitis, where the patients have this continuous amount of pain. Have they come across that?
Okay, so the question was, have we looked at the cannabinoid receptors, CB1 or CB2 receptors, in chronic pain patients, such as pancreatitis and other different pain conditions? And there are some good labeled cannabinoids where you could look at binding and put the patients in to do imaging. As far as I know, that hasn't been done. I know there was interest by Abide Therapeutics to do that, to look at the brain changes. There have been studies largely in laboratory animals that show mixed effects. Like, we found MAG lipase goes up. We didn't find changes in cannabinoid receptors. So it's kind of all over the map. And of course, when you think about pain, it is so multi-dimensional. There's a sensation of pain, and we didn't really get into this, but one thing, one reason why cannabinoids have failed in various pain trials is because the endpoint. They're not opioids. So on a scale of 1 to 10, and if you're going in with like a 7 or 8 severe pain, opioids are very good at bringing that back to like a 2 or a 3 or something. Cannabinoids, not so much, but what they are good at is helping patients sleep, or they still feel the pain, but it's not bothering them as much. Or, I could eat now, or I could get out of bed. So there are other kind of functional measures that they have benefit, but they're not opioids.
Pain issue and global sort of thing that they have to do back.
Right. So they're really, they're more palliative when you think of it. So when you look in the classic, brain regions, and there's been some work showing, again, in like rat models, that some of the circuitry, there are changes of receptors in thalamus. The animals are useful, but they have their limitations, and they don't always translate.
Thank you very much.
Appreciate that question very much.
Maybe we'll just tackle 1 or 2 questions from online. One is about immunosuppression. Is there any evidence of immunosuppression with MAG lipase inhibition, given that CB2 receptors are found on immune cells? Any comments?
Okay, so I think about this more in terms of the innate immune system. So you're reducing a lot of the mediators that cause inflammation and pain. There is other work out there that's looked at, general immune function. So I guess the question really has to do, is there concern that there's going to be immunosuppression and patients could be more susceptible to diseases? And I don't think there's strong evidence for that, but I wouldn't rule it out either. The bulk of it is, anti-inflammation in terms of innate immune responding.
Last question online is about inhibiting MAG lipase and vasodilatory prostaglandins. So could vasodilatory prostaglandins then be reduced and actually worsen blood flow and potentially worsen a sickle cell crisis?
Oh, wow. That's a great question. And that's why we're not going to just jump into any kind of clinical trial. I think any of it's possible. Yeah, do you have there's...
Dilates and it makes it better because basically the sickling happens and it prevents the blood flow. So there is a stimulant.
But it's going to reduce to prostaglandins that are vasodilatory in properties.
Is that what, okay,
so MAG lipase would reduce to prostaglandin expression,
so
therefore reducing those that are responsible for vasodilatory properties.
So is it going to reduce the blood flow?
That's the question.
Well, yeah, if it reduces blood flow, then it may not make it any worse.
So
It's good. It's vasoconstricts of reduces the blood flow. Then you can argue about it. So.
That's a really good question, and I think that's going to have to be empirically answered.
I think so. Well, thanks everybody for coming. Thank you, Dr. Lichtman. We appreciate it.
My pleasure. Thank you for your time.